your favorite pairing. No, not your OTP you've got as headcanon. I mean like a classical combination, like red wine and steak dinner, or the good old peanut butter and jelly sandwich. Think of a matching outfit and jewelry combination, or colors like red and green, or blue and orange. What do all of these things have in common? The parts complement one another. And today, we're going to look at a system which complements our immune system. Welcome back to the science scene. My name is Shelby Bradford and I'm the Geeky Goth, immunology PhD candidate and all-around science freak. Over the last few videos, we've been taking a closer look at our immune system, studying it piece by piece. And it's time to tackle another immuno topic. If you'd like to follow along, like and subscribe to stay up to date on immunology lessons. Over the last couple of videos, we've explored and learned more about the major players and major functions and mechanisms of the innate and adaptive immune system. However, you might have recalled one element from our introductory who's who that we haven't really talked about again. This was because, well, it needed its own video. That is none other than complement. This collection of specialized proteins works within the innate immune system. So if you recall, that means that it'll be activated by general structures shared by pathogens, and that it does not have the ability to improve its response or to generate memory. However, as we'll see, it often relies upon antibodies from the adaptive immune system for its function. We've got a lot to cover here, so buckle in and let's get to it. Now, as a kid who was always horrible at remembering names and dates, I don't often say this, but it will probably be easiest for us to start with the history lesson. Like with many things in science, we learned about complement by studying one thing and making a strange observation. Scientists at the time were trying to better understand exactly how the body established protection. The prevailing theory at the time by Eli Metchnikoff was that all protection was mediated by our phagocytes. This was challenged by a variety of individuals on various grounds, but it wasn't until experiments done by George Nuttall showing blood alone could kill bacteria that was put into it that there was much traction on these other ideas. This concept was further supported when Jules Bourdet showed that if you took blood from an animal that you had immunized against a pathogen and heated that blood, it would be unable to kill bacteria, even though this same blood could be given to a different animal and protect that animal against disease. We would later come to understand that this protection against disease was mediated by antibodies, which we talked about in a previous video. However, the most compelling evidence from Jules Bourdet's research was that if you took that same heated blood from the immunized animal and you added non-heated blood from a non-immunized animal, this mixed blood could kill bacteria on contact and still protect animals. This was an unexpected finding, leading to the conclusion that there must be some extra part of the immune system, something that is complementing these antibodies. Now, if only they had waited to study it a little bit more before deciding on a single name to describe it, because what they were observing was actually many pieces working together. So a big picture overview of what we're about to look at. Complement is an extensive assortment of proteins which travel our blood in an inactive form, usually, until they latch onto a target. This target could be either the back of an antibody or sugars found on the surfaces of pathogens like bacteria. The binding of this initial complement protein begins a series of cascades of more complement proteins binding, cleaving, and activating. All of this serves a few goals. At the site of all of this action, this protein complex will form a unique structure dubbed membrane attack complex. Coolest name ever, by the way. This MAC is going to shove itself into the cell wall of bacteria, forcing the insides to spill out to effectively kill it. Additionally, many of these proteins piling up will be targets for antibodies that will signal to phagocytes, like macrophages, to eat up this monstrosity. Alternatively, these complement complexes could even be recognized themselves. Finally, many of these cleaved off fragments travel to blood as anaphylatoxins. As we mentioned in other videos, these will aid in triggering inflammation and recruiting more immune cells. Now that we've gotten our toes wet, let's dive into more details. Because this system got named before we realized it was actually several proteins, the names are, I will confess, a hot mess. You would not be alone if you needed to get a pen and some paper to make a cheat sheet. In fact, speaking from experience, I would actually recommend doing so. Ready? Here we go. As we said, things are going to kick off by a complement protein recognizing either an antibody or sugars. These differences are distinguished as different routes or pathways to complement activation, and there are 3. We'll go ahead and start with the simplest of these pathways involving antibodies in the activation process, which is called the classical pathway. We usually see antibodies drawn something like this, resembling a Y. The 2 branches at the top are where the antibody will attach to its target, while the stem sticks out to be recognized by other immune cells, or in this case, a complement protein called C1. Once C1 binds, 2 more complement proteins, C2 and C4, are recruited and cleaved to form C2b and C4b, which together form a structure called the C3 convertase. Its job is to split C3 into A and B fragments, with C3b being used to bring in C5 and splice it. This spliced C5 finally will recruit C6 through C9. These proteins form the MAC. Now, if you're curious about those A fragments, they are the anaphylatoxins we mentioned will be produced through all of this splicing and cleaving. Okay, I know that was a lot. Feel free to pause to review, collect yourself, or even go back for a second take of all of that information. Now that you have a handle on the classical pathway, let's explore pathway 2. It's thankfully very similar to the classical pathway. Actually only differs in how everything begins. Instead of C1 and an antibody, the cascade is started by another protein complex in our blood called mannose binding lectin, or MBL. MBL will recognize mannose sugars on bacterial surfaces. After MBL binds mannose in this activation step, the rest of the cascade is basically the same to what we saw in the classical pathway. So this pathway with MBL was dubbed the lectin pathway. 2 down, 1 to go. This final pathway looks pretty different from the other 2 we've discussed, and I'll warn you, may lead to a minor existential crisis. Now that I have your attention, let's jump back in. I told you that these complement proteins circulate our blood in an inactive form. Usually. In this last pathway, the cascade starts by C3 cleaving itself somewhat randomly, and this actually occurs pretty consistently in our bloodstream. If the C3b attaches to a pathogen, it will work to engage factor B, cleaving it to BB with the help of factor D. Now, this C3b-BB complex forms another version of the C3 convertase. It will activate C5 as in the other two pathways, but also consistently cleave more C3 into C3b to promote more complement activation. This is your stereotypical positive feedback loop. Now, beside your brain being a bit boggled from all of this complement alphabet soup, you might also be having a crisis about this sporadic and almost constant activation of this immunological grenade in your blood. But fear not, because this Wild Road show comes with breaks. Traveling alongside C3 and our other fun factors is factor H and factor I. If C3 binds not to a pathogen surface, but to something of ours, then it will get attacked by factor H and, with the help of factor I, rapidly be degraded. This doesn't happen when C3b binds bacterial surfaces because the structure it attaches to on bacterial surfaces blocks where factor H would bind. But if C3b attaches to something on our cells, this portion is open for factor H to bind. So thankfully, we are unlikely to experience mutually assured destruction from these mini-attack arsenals. Now, as with many of the aspects of our immune system, one mishap can lead to dramatic consequences. We haven't really discussed these to this point since we're still just covering the basics, how things should work and work when all things go correctly. But you could imagine, with such a large system and so many moving pieces, that a lot of things could go wrong, and they do. In due time, we can explore these other aspects of the immune system, or when it fails. But for now, that is our exploration into this incredible companion of our immune system. Make sure to hold on to that cheat sheet and check back in later for more immuno adventures and more awesome science.